So in this second part of the Lecture 4 podcast, I'm going to look at proton pump inhibitors and various aspects relating to their interactions with their biological targets, the proton pump, as well as some relevant aspects regarding the biology of the parietal cell. Now let's first look at how proton pump inhibitors reach their targets on the parietal cell. So as explained by Professor Alexander and Dr. de Matteis, proton pump inhibitors are taken as capsules where the enteric coating protects them from the low pH in the stomach. Once the capsules reach the small intestine, the proton pump inhibitor is absorbed and enters the circulation, and so via the blood and the capillaries it reaches finally the parietal cell, where it has to cross both the basolateral membrane as well as the apical membrane before it can actually interact with its biological target. Now, before the proton pump inhibitor can react with the proton pump, it needs to be activated by low pH. So here on the left you can see the proton pump inhibitor, in this case it's omeprazole, and at low pH this molecule undergoes a rearrangement as shown here to form this highly reactive species. So this species is formed at low pH, and when it's in the vicinity of the proton pump, it will be able to form adducts with cysteine side chains via disulfide bridges, as shown here. So this is the S coming from the proton pump inhibitor, and this sulfur atom is from the cysteine side chain. And so these adducts inhibit the proton pump. So this is a more detailed schematic to illustrate how proton pump inhibitors interact with their molecular target. So we've got here blood vessels or capillaries and the parietal cell here, which is present in the gastric pit, in the fundus of the stomach. So as we discussed in chapter 1, when the cell is actively secreting hydrochloric acid, the proton pump will be present at the apical membrane. But also note that some of the proton pumps will be present on inactive vesicles in the cytoplasm, the so-called tubular vesicles. When proton pump inhibitors are taken, they will enter the circulation, so here in red. They will diffuse from the capillaries through the interstitial region, across the basolateral membrane, and enter the parietal cell. And when they cross the apical membrane, then they encounter this highly acidic environment, leading to the rearrangement to form this highly reactive species, which can then react with the cysteine side chains of the proton pump, which are localized in the close vicinity. So the two prerequisites of proton pump inhibition is that the proton pump inhibitor needs to encounter this environment with very low pH to activate the proton pump inhibitor, as well as the vicinity of the proton pump, so that it can react with the cysteine residues nearby. And so those prerequisites are met in the canaliculi of the parietal cell. Proton pump inhibitors, such as here in the case of omeprazole, can enter the parietal cell by simple diffusion driven by a concentration gradient. These molecules are not very polar, they're not charged, so they can enter simply by diffusion. And the same is true once they've entered the parietal cell. They can cross the parietal cell into the canaliculi, into the lumen, by simple diffusion across the apical membrane. However, here they encounter this very acidic environment, and so the proton pump inhibitor will rearrange to form this highly reactive intermediary species, and this species is charged, and therefore it is unable to diffuse back into the parietal cell. So it's trapped in this acidic environment in the canaliculi in the vicinity of the proton pump, with which they will react via the cysteine side chains. So the next question is whether all proton pump inhibitors interact in the same way with the proton pump and inhibit the proton pumps in the same mechanism, or whether there are differences between the various proton pump inhibitors that are available. And so, of course, some of the manufacturers will try to convince you that their proton pump inhibitor has unique characteristics. So is there some evidence for this? Well, it is known that various proton pump inhibitors interact with different cysteine side chains. So if you look in this table, we have just collected some information regarding 5 proton pump inhibitors. Then it's clear that all of these proton pump inhibitors react with a particular cysteine, which is called cysteine 813. So that's just the position of that amino acid. So they all react with this particular cysteine side chain. However, you can also see that most proton pump inhibitors interact, at least to some degree, with other cysteine side chains of the proton pump. And so, for instance, in the case of omeprazole, it also reacts with a second cysteine residue at position 892, whereas, for instance, pantoprazole interacts with a cysteine side chain at a different position, position 822. So what does it mean, and does it explain that perhaps proton pump inhibitors might interact in different ways with their biological target and may have different effects? So here we have a more detailed view at the various relevant cysteines in the context of the proton pump. So here we have the schematic structure of the gastric proton pump that we've discussed in lecture 2, with here this bundle of helices that cross the membrane, and highlighted are the various cysteines that can react with the different proton pump inhibitors. And so this relevant section here is enlarged here on the right. Now all proton pump inhibitors can react with this cysteine at position 813 in orange. And that would lead to inhibition of the proton pump. But in addition, the different proton pump inhibitors can react with other cysteines in the vicinity. So for instance, omeprazole can react with this cysteine here in green at position 892, which would not inhibit the proton pump. Lansoprazole and related proton pump inhibitors can also react with this position, with this cysteine at position 8321, which would inhibit the proton pump. And proton pump inhibitors such as pantoprazole can react with these cysteines here at position 822. Now the various adducts of proton pump inhibitors with these cysteines have different consequences. So if we now just focus on the cysteine 813 and 822, then it has been determined that adducts with this cysteine 813 are much shorter lived than adducts formed with this cysteine at position 822. So that would suggest that proton pump inhibitors such as omeprazole are less effective in inhibition of the proton pump compared to proton pump inhibitors such as pantoprazole, which can react with the cysteine A22. Now, of course, this is just based on the characteristics of binding with the proton pump, and it does not take into account other important characteristics such as the other pharmacological characteristics. So now let's talk about something slightly different. So it is known that proton pump inhibitors require some time before they have achieved their maximum effect. So it may take up to 3 days before they have done that. And this can be understood by the biology of the parietal cell. So as we discussed in lecture 1, the proton pumps are present at the apical membrane, where they are actively contributing to secretion of gastric acid. And they're also present on these cytoplasmic vesicles, these tubular vesicles, where the proton pumps are inactive. And so there's a continuous movement from proton pump inhibitors from these vesicles to the apical membrane and the other way around. Now, as we just discussed in this podcast, the proton pump inhibitors can only react with the proton pumps when they've encountered this acidic environment that activates them. And so proton pump inhibitors can only react, therefore, with the proton pumps that are present at the apical membrane, and they cannot inhibit the proton pumps localized in these cytoplasmic vesicles. And so it will take some time before all proton pumps have been exposed to the proton pump inhibitors at the apical membrane, which explains why it takes perhaps up to 3 days for the proton pump inhibitors to achieve their maximum effect. So finally, I would like to discuss briefly the effect of the use of proton pump inhibitors on the food digestion. So the role of acid in the stomach is twofold. One, it has an antibacterial role, and secondly, it facilitates the activity of the peptidase enzyme that plays a role in the breakdown of proteins. And so this is inhibited, both roles are affected by the use of proton pump inhibitors. So one might expect that people taking proton pump inhibitors have a slight problem eating a protein-rich diet. However, this is not the case, and it can be explained by the fact that the physical function of the stomach is of course unaffected, which is a very important role of the stomach. And secondly, it can be understood by the fact that many proteolytic enzymes that are involved in the degradation of proteins are secreted by the pancreas into the small intestine, and so evidently the secretion of pancreatic enzymes can compensate for the loss of the peptidase activity, in the stomach when proton pump inhibitors are used. Of course, there is some evidence that people taking proton pump inhibitors are more susceptible to food poisoning, which can be understood based on the antibacterial role of acid in the stomach.